Good morning. Today's topic is protein unfolding and refolding. That is, protein, it is going to unfold and after that it is going to refold. That is, first it is found in a folded state, then it will unfold and then it will refold. Then first let us begin with a protein. You all know that what is a protein. Proteins, carbohydrates, fats, they all are the main components in our body. We all take protein in form of cereals or in form of the wherever protein is present. Then we know that all proteins, they begin their existence on a ribosome as a linear sequence of amino acids. And then if we go to the structure of proteins, we have primary structure, then we have secondary structure, we have a tertiary structure, and then we have a quaternary structure. Then what is the primary structure of a protein? It is just a determined amino acid sequence. It is an amino acid sequence only, and when it gets folded, when it forms a polypeptide chain and gets folded, then it forms the secondary structure. Then you have tertiary structure, that is association of multiple secondary structure domains. When secondary structure, they associate, they form a tertiary structure, and then we have a quaternary structure, that is association of multiple folded polypeptide chains, and they further form a three-dimensional structure of proteins. Then after that we come to unfolding. As you already know that protein is found in folded state. After that it is going to unfold, and after that again it is going to fold. That is known as, you can say it, refolding. Folded state, it is the native state and it is catalytically active. So you should remember that folded state is the active state of protein, not unfolded state. When folded state it is going to unfold, it is known as denaturation. And when this unfolded protein will gets refold, this is known as renaturation. But you should remember that a protein, when it gets unfold, it is not going to get completely fold. Somewhere there will be a problem. So you can say it partially folded. For example, if you take an example of spring, you just stretch it, and after that if you are going to unfold it, somewhere there will be a problem. It is not going to unfold the way you are having it. For unfolding you have to just denature the protein. That first heading is protein denaturation. Why do you have to do protein denaturation? You have to denature it to unfold the protein. So how you will denature it? First, you can denature it by temperature. Second, you can denature it by pH. Third, you can denature it by organic solvents. Like acetone and alcohol. Then you can use urea. You can also use guanidine hydrochloride. Now let us come to the temperature, how it is going to affect. If you increase the temperature, then it is going to break hydrogen bonds. As you already know that in proteins, hydrogen bonds, hydrophobic interactions, and then you have electrostatic attractions, they all break when you denature a protein. So when you increase the temperature, it is going to break hydrogen bonds. Then when you increase or decrease the pH, it is going to affect the net charge. And what will happen if net charge is there? Then it will affect electrostatic attractions and further which will disrupt hydrophobic interactions. Then you have organic solvents like acetone, alcohol, then you have urea, guanidine, hydrochloride. They all are going to weak hydrogen bond and hydrophobic interaction. After protein denaturation or the protein is unfolded, we come to renaturation. Renaturation is unfold protein. You have to get it in its folded state or in its native state. But how this can be achieved? This can be achieved just by maintaining the conditions in which the protein is stable. So, you have increased the pH or you have changed the temperature or you have applied the organic solvents, you will just remove them and the protein will get into its folded state, that is catalytically active or that is active state. For example, if you take the example of ribonuclease, you can take the example of ribonuclease. You can see that this protein is in folded state. When you will add urea and mercaptoethanol, this is the folded state and this is catalytically active. You see that this gets unfolded disulfide cross links are formed in cysteine residues, and after that if you just remove urea and mercaptoethanol, it will get into its folded state again. Which is native one. So, you have seen that the urea and mercaptoethanol, they play a role in unfolding and refolding. When you have added, it will unfold, and when you remove it, it will get into its folded state. The unfolded state is characterized by high degree of conformational entropy and relatively high free energy. But in that case you see that there is a collapse state. When you are unfolding it, there is a state called collapse state, and this state is known as molten globule, and there you have a high content of, you are having a high content of secondary structure. Secondary structure is prominent or it is mostly present when you unfold a protein. We can also see unfolding like this. This is the unfolded state. And entropy is highest at this point, and this is the folded state. And somewhere it is collapsing, that is collapsing, we already know that it is known as molten globule, which is having a high content of secondary structure. One of the disease is also linked to protein unfolding, that is known as cystic fibrosis. It also occurs during a problem or during a mutation, during a mutation in protein folding. It is caused by defects in a defect in membrane bound protein that is CFTR, which is responsible for transfer of chloride ion. When there is a mutation in this, then cystic fibrosis is caused, which is a very common disease in humans. It is caused due to deletion of phenyl residue at 508 position, which results in improper folding. So, proper folding of a protein is very necessary, because if there is improper folding is there, then it is going to affect the structure, and during protein unfolding it is going to lose its function. If when a protein gets unfolded, it will lose its function, and when it gets refolded, it comes to its folded state, then it is going to regain its function. So, it is necessary that a protein should remain in native state. When it gets unfolded, it has to refold it to acquire its properties. Thank you.